Hello, everybody. We are live with the after show. I am here with Nesslig20 and nobody else. So let's see. Finding Truth has the link, and he said that he would be here. So hopefully he's here soon. A few other people have the link, but are also not here. So, Nesslig, what did you think of the debate?
Well, speaking about like in terms of interrupting and like it wasn't a train wreck to say the moderation was good and everyone has at an equal time to speak and such. Oh, I get the feedback from Ahmed, but I think the topic was a bit ignored. Like it was really quick to
Hey,
change
Ahmed.
the topic from the Cambrian to something else instead. So I was hoping that the topic was more focused to be on the main topic. That's my assessment.
And Ahmed, what do you think? How do you think the debate went?
Well, yeah, I think I think the main argument there, the thing that makes me really interested about the Cambrian explosion was the explosion of information. So I really wanted Jackson to take a specific position about what he thinks this is coming from, and then we move on. I think he dwelled a little longer, or for the whole thing, I think, on the point of whether how to measure information, etc. While the examples were so clear, and he actually agreed that there is extra information. So I don't know
Well, he
why he
agreed
was
there was
so
different
resistant about it.
information. He agreed there was different information.
Yeah, he agrees that you can perhaps describe the parts, but like how you connect that to information, or like you say you can measure information in terms of tissue types and also cell types, and perhaps even more different means, but how you connect all those things to quantify information is a bit fuzzy, to say the least.
Yeah.
Like for example, if I have one organism who has many cell types, but a few tissue tissue types, but another organism who has a lot of tissue types but a few cell types, then how do I say that the one is more complex than the other, or how do I make that judgment?
Well, you make a judgment because if you're comparing two things, you have to pick up a common denominator to compare them, you know.
Right.
So
you need many organisms. You cannot do that. Like, how can I compare, like for example, the amoeba to a human? Like the amoeba doesn't have cell types because it's just a single cell. At least, like there are amoebas who aggregate to like a complex colonial organism, but even single-celled amoebas can have lots of, like 4 times as many genes as we have. So in that sense, they are more complex than us, but in another sense, they are less complex than us. So it becomes really arbitrary to say one has more quote-unquote information than the other.
Well, I don't think the size of the genome is can be used as a measure of complexity, because
that's a problem. Like, why do you exclude that metric from the information discussion?
Because we really don't know how the genome is interacting with the rest of the cytoplasm to produce what we see as a complete organism. And I think other biologists also. I was listening to something from Charles Marshall yesterday, and he was saying more or less the same thing. You see, the problem is, I can appreciate why this problem is coming. For example, if I am giving you a data file that is compressed, okay, with some compression software, and another one that is not compressed, so the uncompressed one will have a much bigger size, yeah? The compressed one will have a smaller size, but essentially both of them are having the same amount of, they're actually the same file.
Yeah, but in information theory, the definition of information is pretty clear. Like like it's Shannon information, right? So I have either a 1 or a 0 at a certain point that resolves a question of uncertainty, and that is the information that is defined as a bit, if I am not mistaken. Because it is not my area of interest. I have some basic knowledge of. I don't know if Dapper knows more about this. Am I right about saying that 1 bit of information is like a 0 and a 1?
Yeah, so that is the sort of the Shannon definition, and you can measure many kinds of information using Shannon information by determining how many possible characters there are in the signal. So, for instance, DNA you could represent as a 2-bit information system, because with 4 base pairs, you can assign 1 as 0 0, 1 as 0 1, 1 is 1 0, and then the last one is 1 1. And so you could take the entire genome, multiply the number of base pairs by 2, and that would be the smallest way you could represent a genome unambiguously.
Yeah.
In terms of bits.
Yeah. So let me take this example. If you have a screen and the screen is completely green, yeah, and the screen is like HD, so it has plenty of pixels. So one way to describe it using a bitmap file would be that it has 124 by 768 bits, okay? And this is plenty of information, right? And another way to describe it is to take an image compression system, and it will tell you there are that much megabytes of bits and all of them are green, and this will be few bytes of information.
Yes, that would be a very easily compressible picture.
The same amount, yeah. They're both conveying the same amount of information. And the trick here is that there is another layer of information, which is the compression system, right? And this compression system here, now you have to add the information that you used to design the compression system about how to make the compression to the information content of the compressed file.
Okay.
So my argument here is we do not understand how the genetic system interacts with whatever else is in the cytoplasm or even the cell membrane to produce the final creature. To the extent we do not know, for example, if something like the human genome has a level of compression in the genetics, which is then decompressed by epigenetics or ontogenetics or whatever thing that controls morphogenesis.
Well,
so since we do not know,
the genome never shrinks in size. Only to then grow back to its original. So the genome is never compressed into a smaller size, only to then be re-expanded. I'm not sure how you're trying to map compression of data onto the genome.
I think you're using like the term compression differently in like we have. Of course, there are mutations within different cells in our body, but the genome stays pretty much the same size regardless of the different cell type. Do you mean like gene silencing? Like in some cell types, like for example, the insulin gene is not expressed in most of our cell types, only in the specific cells of the pancreas. Are you referring to gene data compression in those terms? Like I don't know what you mean by data compression in that regard.
So for example, you can have one gene that expresses a protein of that structure in one way, or it can express the protein that then gets excised and it becomes a different protein because some other epigenetic mechanism has taken this decision.
Do you have an example of such a gene?
Hence, it is not that I don't have an immediate example in my mind, but I'm sure I can find one.
Well, that would be an interesting thing to find because I'm not aware of any such genes. But then again, I'm not an expert on genetics. So, but to be honest,
are you referring to like alternative splicing or?
Yeah. So in many cases, I can't remember one specific example, but I think we can have a look at that, and maybe we should have some discussion about what's really going on.
But I'm also like doubting whether you can say like, oh, because we don't know how the genome affects the organism, therefore we shouldn't use the genome as a metric, which is basically saying like, oh, we cannot apply it, so we better ignore it. Like, but it's still a bit of an arbitrary. Like if it is important, then we should be using it, right, or not?
if eventually we have a system, it is like the example I was given, but I was cut short on that. If we take all the recording that we are doing now, the image can be an example. Another obvious example is that you can record all my voice in a WAV file, and that will be quite big. But if the information content you are interested in is what I have said, then the transcription of that into letters will be much smaller, yet conveying the same level.
But now you're talking about like semantics, like the meaning behind the information, and that is something that Klaus Shannon deliberately avoided. Like it's very problematic to define information as in semantical, like when you talk words and such.
Yeah, I think I could give an example, right? So when we're talking about Shannon information, which is one of the only ways that we have to quantify information, right? We can do things like compress images. So if I have two images, right, and they're both the same number of pixels in the X and the Y direction, and one of them is one of those chess problems. You ever seen those in like the paper where you have a chessboard layout and the question says, you know, white to mate in 3 moves, and you have to figure out what the 3 moves are that gets white to checkmate? Well, that kind of picture compresses down to a very small file size because it's mostly composed of big blocks of a single color. It only has 2 colors. You can compress that down to a very small file size. But if you have another picture of the same size in which every single pixel is simply a randomly assigned color, so it has random red, blue, and green values, that is essentially uncompressible. Now, the thing is, to humans, we have...
That's basically static. It's basically a static image.
Right. Like, so the static image, which to humans means essentially nothing, because there's no puzzle there, there's nothing for you to, you know, get your brain into, there's no pattern, there's no nothing. It's just a bunch of nonsense. That has a lot more Shannon information than the chess problem, which is going to have extremely low amounts, which is one of the reasons why your intuitive human idea of how much information is contained in a thing doesn't really matter that much to science, because in science, information is all about what does it actually take to get whatever particular signal it is across. And it doesn't matter whether or not that signal is meaningful to you as a conscious human. All that matters is that the signal be reconstructible. And it also doesn't really care if the sender or the receiver understands anything that's happening, or even if they can understand things. Dumb machines can send and receive Shannon information just as well as living, breathing, cognizant humans. So using this idea of, well, you know, we kind of feel it in our bones that this is more complicated than that, doesn't really help in science, because one of the things that science has taught us is that human intuition is absolute garbage. There is no place for it in science. It is completely worthless on anything other than the most mundane of tasks.
Yeah, it reminds me of like one of my favorite Neil deGrasse Tyson quotes: "The universe has no obligation to make sense to you."
Right. So that's one of the reasons why I think we're trying to get a more rigorous idea about information from you.
Yeah, like I would have. Oh, go ahead. Sorry.
Yeah, I want to hear from Ahmed.
Yeah, so that's why I wouldn't really count on human intuition on this, and I would not really also count on an obliterated understanding of the genetic system. And I would count on something that is very easily observable, which is the resultant of that information, which is essentially the organism. Right. So whatever the genome is that produces an amoeba or an E. coli is obviously much less information bearing than the genome that produces a human being.
But it's not obvious.
Obviously, you say you want to lie to intuition, but when you say obviously, then you are a bit of like appealing to our intuition.
Yeah, see, this thing. First of all, I don't actually think it is obvious, and even if it were, that would still just be intuition.
Yeah,
I'm appealing to observation. You look at something, just like the example of the bicycle and the car, or like the example of the screen, or the image on the screen. If whatever is manifesting as a single green screen, one color screen, versus a screen that has image of flowers and animals, you do not really need to look at the file. If you look at the screen, you know that this one has less information than that one.
But the thing is,
you essentially observe it.
There are other things which I could look at that would not obviously to my eye contain less information, but which in fact would. For instance, let's say I just have.
Yeah. So
let me give my example.
In the backdrop of that.
Ahmed, I need to finish.
Right. Go ahead.
Okay. So for instance, let's say I have 2 pictures on my screen. Both of them are gradients from blue on one side and green on the other, right? One of them is a PNG file that's an 8-bit PNG file. We'll go 16. So we got a 16-bit PNG file going from blue on one end to green on the other. But then I have a 32-bit float TIFF file with the same gradient. To my eyes, those are going to be essentially identical. But that TIFF file actually has a whole lot more information because it's using a 32-bit float instead of just a 16-bit color. So that TIFF file is going to be a lot bigger, and the gradient is going to be smoother if I were to zoom in on it. But from just me sitting at my computer, I usually couldn't tell a difference. You cannot use human intuition to determine what things have more information. You actually have to dig into it. You have to take numbers and measurements. This is one of the reasons why we ask you, how do you measure it? What units do you use? Because science isn't about, this feels like it has more information. It's about, here's the measurement I took. It has this many bits of information. Here's how I know. That's what we would like from people who use information as an argument.
Because they're using the information argument incorrectly, in my opinion. Because if you have a transmitter that is transmitting ones all the time, information is also a measure, the information content of a message, or the next bit in a message, or the next piece of information in a message, is also dependent on the level of surprise that this message brings.
That's true.
This is actually the terminology used in the theory.
Yep, if it's always one, that signal actually has no information.
Yeah. Yeah, if you have something that is all the time broadcasting one, then good luck. This is not really broadcasting that much information.
You can just contrast information to repeat it.
Right. Well, I mean, there's real-world examples of this. For instance, like, let's say you're trying to transmit a message in English using ASCII, right? Well, the first few bits in every character in ASCII are always zeros. So you could compress that signal by cutting out the first few bits because you don't really need them. And that would, in fact, give you the actual amount of information, because then your chances of getting a 0 or a 1 are always about the same, basically 50-50 for the entire message. Now, it's not actually because English has, particular rules, like E is the most common letter, and certain punctuation marks come after certain strings of, letters. But at a first level approximation, we could compress it that way. But the thing is, if you're talking about the information in the genome, and you want to talk about things like compression, or if you're talking about information in terms of morphology, how are you actually going to measure it? Because if you want to say there's new information, or you want to say this has more information, or there's been an injection of information, you need to be able to quantify the information and tell us where the new information is that you think is new, and why it is that this is some kind of problem or, say, mutation. Like, you have to start off, the very first thing you have to do to get this kind of thing off the ground as a science is you have to be able to find an objective way to measure the thing you're talking about.
Mm-hmm.
But we don't have that from you, and that is concerning.
So this was the whole point of my argument, actually, that you do not need to do that. Because why you do not need to do that? Because you are not, there is a big difference between measuring an absolute quantity and a comparative quantity, okay? So if I am to measure my weight versus the Dapper dinosaur weight, okay? So I'm getting the Dapper dinosaur, and I will stand on one hand of a scale, and he will stand on the other arm of the scale. And we will hang to those two arms. And those two arms will not tell us what is exactly my weight and what is exactly the dino's weight. But it will tell us who is heavier. So when you are doing comparative measurements, you do not need to have an absolute measure. You just need a common denominator.
But the thing is,
and this is the whole thing that I was talking about. Yeah, do you get my point?
But there's a problem there. And first, I have a $2 super chat I want to read real quick. So this is from No Last Name, and he says, optical illusion debunk intuition, the vase or two faces image, which I think we are familiar. I think most people are familiar with that. There's the picture where at one point, if you look at the white as the positive space and the black as the negative space, it looks like a vase. But if you reverse it so that the white is the negative space and the black is the positive space, then it looks like it's two people facing each other. Or some versions of it, they are actually like close enough that they're kissing or something, whatever. Anyway, so the problem here is that when you take a look at a scale, there's an objective, one side goes down, the other side goes up. That's not anything to do with intuition. That's nothing to do with intuition at all. The problem is that when you say this thing has more information than the other thing, people aren't all going to agree with you. And what that means is you don't have an objective thing. If you have a scale, in every single case where you put two objects on a properly balanced scale, everyone who watches will be able to agree this one is heavier than that one. That is not the case for information content in animals. Therefore, you cannot use that analogy. You still need to find a way to get everyone to objectively agree, even with your relative measurements. And since there's no way to get people to agree just by intuition, because they're not going to, I guarantee you, then there is no way for you to make these objective comparatives. You need to find a way to measure it if you want to make it objective. We have a way to measure weight. It's a scale. That's great. That's an objective mechanism. I can build one. I can test it. I can take two things of known weight and check to see if they're the same weight. If I already know previously from other scales they are, I can test my scale. Everyone who looks at the scale agrees. You don't have that with information on organisms.
So here is my response to that. What we were discussing there, the example of the eye, or the example of the bicycle and the car, and it is irrelevant that the bicycle and the car are non-biological because we are discussing how to assess design information. So let's say we have a bicycle and we have a motorcycle even to make it easier.
Okay.
Obviously, the extra component here is the motor. Okay. So it is obvious that the information for building the motor has been added. Okay. Oh. So this is exactly, you do not need a fair or an unfair observer to acknowledge that there is a motor that has been added, and that this motor needs information. So this was exactly what I was saying.
But here you're going again across the same problem that you are again appealing to intuition. You again say obvious, but how much information does the bike have, and how much information does the motor have, and what is the difference between the amount of information between them? How do you determine that? That is still how do you calculate that?
And before we go, I want to say that we have hold up, we have a $9.99 super chat. This is from Vandalia 1998, and he says, before I have to leave for work, I just want to say I liked how Jackson brought up sauropods in my giraffe question because I never thought about how bad their neck problem with the recurrent laryngeal nerve would be, or could be before. So yeah, it is, actually it's for that reason, most paleontologists have concluded that sauropods probably had very, very limited capacity to communicate via sound because giraffes already do. Giraffes basically make a few bellows and that's about it. Whereas, other artiodactyls often have quite a few calls that they use to communicate, and it's likely that sauropods would have been almost completely incapable of doing anything other than some kind of rumbly noise with their vocal box, because the recurrent laryngeal nerve would have been, 60 feet long in some cases.
Yeah.
Yeah, and your nerves do not. They transmit signals pretty fast, but there's a noticeable delay even between your brain and your foot. And think about going across down a sauropod neck around the aorta and then way back up to the voice box. It would take forever. So just want to get that. So sorry, Ahmed, you were saying?
Yeah. So I was saying that whatever position you end up taking regarding this information thing, I was really glad that Jackson takes one specific position, and then he would essentially face the same problem. So I offered 3 positions. The first one, tell me, okay, Dickinsonia had more information than trilobites, and trilobites just expressed some of Dickinsonia pre-existing DNA, and then what you will be implying is that the information gradient is reversed.
I don't think he was saying that, but anyway.
No, he didn't. He didn't. He rejected that one, which I was okay. Then he essentially, by rejecting that one, he immediately acknowledges that trilobites have more information than Dickinsonia, because I gave him the alternative proposition.
Well, I don't think he was saying that either, but yeah.
Yeah, he was left with 2 propositions. The second one would be that, no, trilobites exhibiting those new novel complex structures and body plan and organs and systems exhibit that because they have more information. Or number 3, both of them have information of their own. And in the first one, we have a problem of a reverse information gradient. In the second one, we have a problem because now obviously the trilobite has more information, so where did it come from? And in the third one, it doesn't make a difference which one of them has more information, because now there is still an significant amount of information in the trilobite to specify all those extra systems. So bottom line, irrespective which approach that you take, and there is no fourth approach, it's either bigger here, bigger there, or both of them have information of their own. At the end of the day, there is infusion of information all over this Cambrian explosion thing, and it is massive. And the question is, where is it coming from? So this was the whole point. You have new plans, new phyla,
guys. I want to read a super chat because these are much less informal. So I try to read super chats as they come in, more or less. So this is from Rodent No Last Name, who says, "Does the bicycle or car contain the information self-replicate? Are either of them able to pass on information to their offspring? Of course not." I don't know if you guys want to address that, but I'm obligated.
I don't think it has much relevance to it anyway. Yeah.
So I guess.
Do you want to respond to that, Ahmed?
I mean, I do certainly think that is one of the reasons why it's always difficult to make analogies between man-made objects and organisms. But yeah.
Although, in my opinion, that is not the sole the most problematic about the comparison. There are other problematic issues about the comparison. But anyway, I think like Dapper and I have repeatedly expressed our doubts about your definition of information. But when you respond to our questions, you basically argue as if the definition is already a given, and you continue using the word information without actually telling us exactly what you mean by it. So it's still.
So information in this context is what you need to specify so that the morphology can be implemented, which I have actually given an example in the debate that as engineers we do all the time. When you start by the end product, for example, the Japanese did it big time after World War II to raise from the dead, and the Chinese did it after them, and the Taiwanese, etc. And many nations did it, right? They work on re-engineering until they have their own engineering. And what you do when you are reverse engineering something, you take the final product, you reverse specify it, and then you start building it. This is how you do it. And even about the question whether cars or bikes are self-replicating, I can give you a thought experiment where we are talking about a program, a CAD program that generates bikes, and a CAD program that generates cars. I mean, really specify them to the nitty-gritty nail and pipe and materials and bills of materials and assembly instructions and everything. Which one would you think has more information? The one for the bike, or the one for the motorbike, or the one for the car?
What if I said that the bike contains more information? Like, how would you show me that I'm wrong?
I will immediately show you.
You continue to say, "Oh, it's obvious," but then we are talking about something that isn't obvious. Like,
in this case, since you are using a CAD program, and it is the same CAD program, so information density and information efficiency is the same, you will immediately see that the specification file for this one is the least one, and then the motorbike is the next, and then the electric car or the car is the next.
Okay, so
and if you see that one of them has a bigger specification file, while it exhibits less engineering details, then somebody will have to go crazy, and it will never happen, of course.
So your definition for information is how big a file it would take to 3D model an organism.
In this example, in the example that we were taking there, we will take if two organisms are multicellular, then we will take something that relates to them, number of cell types, maybe numbers of proteins. I don't know.
Would the 3D model also contain everything at the molecular level, like the amount of DNA and the amount of protein and biochemistry, like all those things too?
Which example are you referring to?
Like all of it, like from the DNA to the biochemistry to the if you build a 3D model to like the data needed to build a 3D model of the whole organism, do you also include everything from the cell, from the molecular level to the whole organism.
Well, this is the whole problem. We do not have this kind of information to be able to specify organisms at molecular level to this extent. We will be essentially specifying them like we specify bitmaps, and we will have to do a complete set of specification.
But that is the exact problem that we are trying to point out when you use information to describe this.
Because we cannot build them bottom up. In this case, we will have to reverse engineer them, and then we will reverse specify them.
So hold up.
Yeah,
this is the I have a question.
Yeah.
Since it would take a whole lot more to do a detailed 3D model down to what I would presume would be the minimum level, would be the cell level of, say, a whale than a human being, do whales have more information than humans?
I don't know if whales have more information than humans or not. It depends on what systems they have.
Well, they have the same systems as humans have. They have the same tissues, and they have the same organs. But it would take a whole lot more work to 3D model a whale at the cellular level than a human.
And what if you compare it to humans with giant sequoias?
That would also take a lot more work. Actually, you can't even use X-symmetry.
And especially if you also consider the biochemistry, like in terms of biochemistry, plants can do way more things that we can. Like they can photosynthesize, for crying out loud. They can turn sunlight into food, and they can also make certain amino acids that we cannot. We have to eat those.
So, given your method, which of the following three organisms has the most information, and why? A giant sequoia, a humpback whale, or a human?
Yeah, so you will have to pick a metric, like cell type, systems, organs, whatever.
No, we're using the one that you just said. Where we're going to take a CAD program, and we're gonna 3D model it at least down to the cell.
I will have to 3D model it. I don't know, because there will be plenty of fractals there. Yeah, you know, if you want to 3D model something, it will be about the level of detail, not the size.
Well, since all organisms are reducible to cells, let's just say we model it down to the cell size. We model each cell at its appropriate shape. And then, now we have a 3D model of every single cell in a whale, every single cell in a giant sequoia, and every single cell in a human being. Which of those three has the most information? I'm pretty sure it's the giant sequoia.
I would also,
I don't know, and it doesn't.
I would also bet on the sequoia, but that is my opinion, and that's a bit of an intuition, of course.
Right,
to say that the sequoia is the more complex.
Are sequoias the highest form of life on the planet?
So the trick here is actually we had gone through that in the debate, because we are looking at infusion of information. Then we should be looking at the same lineage. You are looking at that precursor and then to the next one. And
well, that is the same lineage. That's all the eukaryote lineage.
Same
this is well within the eukaryote lineage. So there you go.
No, not according to the phylogeny chart that the theory does, because there is a branch point where whales go somewhere and humans go somewhere else.
Yeah,
so
but before
it doesn't make really a difference.
But before that, we're in the same lineage.
Fine, then pick points on the same lineage. Pick points on the same lineage, not branch out. And then I don't know what is according to the theory, whatever is the precursor of a whale, and then you will see if there is an increasing gradient of information. Then you will have to ask the same question again: where is the information coming from? It is essentially the same question since starting from Luca until any extent or any rim part of the tree. Well, doesn't make a difference. But we haven't been able
to figure out if there's an addition of information in the first place, because we haven't been able to figure out what has more information than something else. So, for instance, does a humpback whale have more information than a hippopotamus or their common ancestor? Since hippopotamuses and whales are sister groups within Artiodactyla, I mean, how do we know? They're the closest thing to a hippopotamus is any given whale, and the closest thing to whales as a whole is the hippopotamus.
Also, Ahmed, I think you are asking us a question that you should be asking yourself, not us. So that's all I'm saying.
No, I'm because I said that I am equally okay with any of the three answers to the question, If you are confronted at a point where you say that I don't know, you consider a rhino a precursor to a whale, and you find that the rhino has more information.
No, not a rhino.
Hippopotamus, sorry.
Rhinos are
a hippopotamus.
Yeah,
sorry, a hippo. So if you take a hippo as the precursor for the whale,
which we're not,
or a hippo,
they're sister groups,
and you find that the hippo has more information. Fine, no problem. This is this is position number 1 that I suppose, and this is not a problematic.
The question I'm asking you is, how would we find whether or not a hippo has less information than a whale? If it does, how would we tell? You
will note in this case that this is exactly why the Cambrian explosion is a very nice example, because you do not have to go into this confusion. It's like the other question that came: is somebody with Down syndrome having more or less information?
Okay,
we are not into the very small detail here. We see a very simple creature morphologically and a much more complex creature morphologically, so we don't have real doubts.
Oh, okay, well then,
but when you see creatures that are equally complex,
you're begging the question based by saying, oh, they are simple and they are complex after. So like, but of course we still need to determine whether they are actually simpler before and after. Like,
how do we know?
You haven't defined it.
Right. How do we know if hallucigenia is more complex than opabinia? I don't know. I mean, seriously, how can I possibly know which of these two is more complex and which of them had more information? I don't have a way to assess that. And for $5 we have from Rodent No Last Name, do you not realize that your definition of, quote, information keeps changing depending on context?
Oh, that is also a really good point, yes. It's
No, I don't find that my definition of information changes. My definition of information is very simple and straightforward. Any exhibited or manifested design has information behind it. Now we are standing in front of a very simple question. Whenever you see a new design, whenever you see a new artifact that is exhibited that was not in any of the previous precursors, then you need to ask the question, where is the design information coming from?
I would argue that has never happened.
It's a very simple question. Either you assume it was there all the time or not. If it was there all the time, then the gradient is reversed. If it was not there all the time, where did it come from?
But do you have an example where evolution claims that something came that was brand new and didn't have a precursor?
We were actually discussing that in the debate, and we took the eye of the Trilobite as an example. So the previous thing that we have is a photocell, or a photosensor. Okay.
Well, eyes are.
Then we have a complete eye. So obviously there is extra design.
Well, so we have.
It's undeniable.
We have things between image-forming camera-type eyes and simple photosensitive eye spots. So this is, again, we have precursors for this. So I don't see what your argument is here. And even if we didn't have extant and or fossil examples of precursors to what we have in some organisms now with camera eyes, evolution still doesn't claim that such things didn't exist. So even if we were completely ignorant of the morphological development of eyes, that still wouldn't be evolution saying that they simply formed out of nothing. So your original point here was to say that we need to have a mechanism by which truly novel things without precursors develop. But what I am saying to you is, evolution doesn't claim that such things happen at all. Every development in evolution works off of the precursors that were there in ancestral populations.
Yeah. But at the best example of an innovation would be de novo genes, but even those, they came from like a segment of non-coding DNA.
Excellent. So within the very relatively small time of the Cambrian explosion, we need to understand how such a huge amount of information that creates an eye, a compounded eye, from a very simple photosensor.
Well, again, you say a huge amount of information. How much information are you talking about?
Information that is enough to specify an eye, a compounded eye, the nervous system that captures the images for it.
Yeah, but how much? Like you say huge, but how huge?
I don't know. I know how much information that we need, quite big, to create the counterpart electronic part.
You say you know how much it is. That's great. So if you know how much it is, give me a number and a unit, and then justify your measurement. Otherwise, you can't say you know how much.
So
the next thing I need to hear from you is a number and no. The next thing you need to say is a number followed by a unit. Do you have those?
Dapper designs are not measured by numbers.
So then you don't know how much information it has. That was a lie.
There is nothing on the face of the earth called give me a number that corresponds to the design of a car.
Which is why people don't talk about how much. Which is why talking about how much information is in there is silly, because we don't measure designs in terms of information.
You say there is a huge amount of information, yet you say it's not an amount at all.
Right, amounts can be measured.
You're contradicting yourself.
No, I'm not contradicting myself. If I want to build a digital camera now, and the system that stores that image and the system that processes that image to identify that what's in the camera is a cat, okay? So you can quantify that by the electronic circuitry and how much specification you need to make for that, and then how much information or how much you need to do in terms of programs and how much you need to do in terms of a neural network, and each of them will have its own measure.
Okay,
do you have at the end?
Do you have any measurements for the eye?
I'm sorry?
Do you have a single measurement that you can apply? Do you have any measurements that you can apply to the information required to go from an eye spot to a compound eye? A single measurement for any of it. Do you have one?
What do you mean by have one?
Okay, when you were talking, okay, so when you're talking about a camera, right? If I need to build a camera, I can give you a whole bunch of measurements. I can tell you, okay, how much of any given particular raw material do I need? Like how much silica do I need? How much iron do I need? How much manganese do I need? I could tell you, okay, how many transistors I need, or I could tell you how many chips of certain kinds I need. I could tell you how big the software files are going to have to be that run in the camera to actually take the information in. I could tell you things like how much mass the camera is going to have, something like that. Like these are all measurements. I can give you numbers and units. Do you have any?
Yeah, but
let me finish. Do you have any number and unit that in any way relates to the information required to go from an eye spot to a compound eye? And if the answer isn't yes, followed by a measurement, you have no point here. Because you're not talking about anything that you have. So yeah, do you have such a measurement?
Exactly as you don't have let me give you a full answer, like the full question.
Sure, go ahead.
So just exactly as you don't have a single one number measurement for how much design information is in a camera, you will not have the same in an eye. But in a camera, you will do the corresponding to an eye. In a camera, you have a lens. You have information about the curvature of the lens, the material of the lens, the focal length of the lens, etc., etc. And these will be specifications that relate to optics. Okay, and then you will have something that relates, for example, if this is a digital camera, to the CCD material, how many transistors, what is the material of the transistors, what is the photosensitive chemical that is used, or what is the photosensitive mechanism that is used to capture the light. And then you will have other specifications for the conveyance of that thing into electrical signals, and how much memory you need, and what are the materials to build the memory, and what are the types of the transistors to build the memory, and what's the software that's managing the camera, etc.
Okay, so
likewise, you will do the same thing for the eye.
Give me one of those measurements.
So, for example,
here we go.
Exactly. So, for example, you are having something like an eye spot in an amoeba, where you are essentially specifying a certain maybe protein or a certain, I don't know what is the active material there. So you will specify a protein. That's what you need. And then you will see what is the chemical messenger that will be turned on or off when there is light, and then when there is no light.
Okay.
But if you are going to an eye, a compounded or even a single chamber eye, then you need to specify the lens of the eye.
No, hold on.
We're
now shifting away.
The curvature of the lens.
You gave us one measurement. We have a protein and a mechanism. Now, to make a camera eye, I'm going to point out we don't need extra proteins or transmission mechanisms except maybe neurotransmitters. So is the added information, in the case of the proteins, this is one measurement that we can actually do. We can do a measurement. We have a protein. Now I need a way to turn that signal into a neurotransmission. So I need to evolve at least one neurotransmitter. So is the amount of information needed in terms of proteins to go from an eye spot to an eye +1 protein. Is that fair?
Do you believe that? Do you believe that?
Well, no, that's what I'm asking you. I'm trying to get some kind of way where we can take your claims and make them objective and analyzable in a way that is actually scientific.
If, for example, you take that, and if we take it that we have a single protein that is photosensitive in the eye spot, then we have one or more, probably more, proteins to create a lens. So this is one protein.
Okay, but that's a different measurement.
To create whatever.
That's a different measurement. We're just talking about photosensitivity as an aspect of the camera. We have one photosensitive protein, and in order to take, just in terms of transmitting the light signal, we need one more protein in order to make it into an eye. Would that be fair in terms of transmission of the information about photoreceptivity?
Of course not, because this is not the eye that we're talking about. We were talking about something that we call an eye that is just on and off. There is light, there is no light, and another one that will give you a shape. That will give you what is your environment.
What extra proteins?
And this is not a single. It's not +1. This is definitely not +1. This is proteins for if we are just taking the measure of the proteins only.
Okay.
So you have proteins for the lens.
Wait, hold on. No, we don't need. We do not need a lens. We don't need a lens.
Why?
Because you already said that you already said that the Nautilus eye is an eye and it doesn't have a lens, so we don't need a lens.
No, I said it's a single chamber. It's not compounded. It's like the human eye.
Well, compound eyes don't necessarily need lenses either, because they can form images based on having pinholes, and then knowing which ones are lit up will then result in a relatively low resolution, but an image. So we don't need lenses to make eyes that form images. So who cares how many proteins it needs to make a lens? You don't need it to make an eye. All you need to make an eye is a pinhole camera, which means you need one light receptor protein. You need a way to transmit it to a nervous system, and that's it. Everything else is just a change in the shape of the already existing body structures. And that is not a protein matter.
No.
That's to do with developmental genes, like Hox genes.
Do you really believe that to build an eye, being a compounded or a single chamber eye, you need to specify two proteins? Meaning that a neural receptor or a neural fiber is made of one protein?
I'll clarify. We need one more protein than we need to make an effective photosensitive spot on a single-celled organism. We need a way, so we already know that rhodopsins in single-celled organisms can signal internally in the cell. The only thing we need past that is a way for that signal to be sent to other cells. That's at minimum one protein. Now, it might be at maximum more, but we need one new protein to get the signal out of the cell. And we already get that before we get eyes, because flatworms are already doing that. So really, to go from an eye spot like a flatworm, which isn't an eye, and I think we can both agree, to get from that to a pinhole camera, which is an eye by your definition, we need literally not a single new protein. Not one. No new proteins are required.
At best, you need the protein. Maybe you need the protein to as a morphogen, maybe.
Maybe, but we also don't necessarily need morphogen proteins to change the morphological structure. That can be accomplished just by things like Hox genes signaling in different patterns. That's all we need. We can get more. We can go above and beyond the minimum. But the minimum to go from a flatworm eye, the minimum to go from just a single-celled eye spot to a flatworm is basically just a neurotransmitter. The minimum to go from that to an eye is no new proteins, only changes in the shape of already existing tissue, which is generally not controlled primarily by proteins. What do you mean, no? A monochromatic,
as you are saying.
A monochromatic. Okay, explain to me how you think a monochromatic, like a monochromatic vision, pinhole camera eye works. How do you think it works?
So let me share my screen, please.
Oh, sorry. Is that not turned on? That should be turned on. I didn't turn anything off.
I think the point Dapper is making is really simple. Like you have on one hand a flat surface, like just an eye spot, and on the other hand you have a pinhole camera, and all you need to do to change one to the other is just make a dimple and then close it off into a tiny hole, and then you have a pinhole camera.
Oh, I think that should be working now.
Yeah.
All right. So I didn't have to.
What do you see?
I see some lenses.
This is lens. Yeah, Trilobite lens.
Yep.
You see Trilobite eyes. So here you see.
We weren't talking about that. We were talking about the pinhole cameras.
What it takes. Yeah. So
okay. So this is
in the debate we were talking about.
Once we get right now, this isn't the debate.
Give me an organism that had.
I'll tell you what.
If you want to,
I'll tell you what, Ahmed. Here's the deal I'll make you. After we exhaust pinhole camera eyes, then we can start talking about lenses. That would be great. I'm absolutely completely on board.
What is?
I also have to read this.
Creature that what is the creature that has a pinhole camera eye?
I have to read this super chat.
I
have to read the super chat. This is $5 from Rodent, the last name. He says, again, your definition of information keeps changing depending on context. Do you understand why that is a problem with your argument?
You need me to answer this question?
You don't have to if you don't want to. I have to read it because I was paid to.
No, I can answer it. So, again, we are discussing how a specific morphology needs to be specified. And the only way it can be specified is through information. And this information, regardless if it is a photosensitive protein or it is whatever we will agree to be a pinhole camera, whatever that means, or a compounded eye or a human eye, it needs information to be specified.
Okay.
It's very simple. Or it's the same thing. What you need to specify it is how it consists of what are the building materials, what is the building materials that you need, those building materials being chemicals or cells or whatever they are, how you will arrange them, how you will arrange other parts of the systems to interact with it so that it delivers the function, and the extra information on top of that will make the organism understand how to use it and make benefit of the information output of it, which is the exact 4 levels of information that I discussed in the debate. So if you are going from the pinhole, I don't know which creature you are referring to, but let's find a creature where we will say this is a pinhole camera.
Okay,
and this means that you have a chamber. Yeah, you have a chamber that has an aperture or something kind of lets light in. You will need to have a membrane there because you will not have whatever the external environment flowing in.
No, actually, you do have the external environment flowing in.
Whatever. I mean, just let's pick up a specific creature and see how it is constructed. You will then need to say how this will be constructed in terms of morphogenesis. It will need to be specified there. You will need to see how to take the neurotransmission and how to make something corresponding to it in the brain.
Right. Maybe we need to back up.
The creature will be wired to understand that this is now an image.
Maybe we need to back up because I think we're now getting into how embryology works, because I feel like you probably don't have a good grasp on that. Not that I'm anything like an expert, but Ahmed, let's say that we have a new zygote of any given species. It doesn't matter. It's some animal, right? Let's say that, just to make things simple, we're going to go ahead and say that this is going to be a, we'll say it's going to be an earthworm, right? So there's not a whole lot that's going to happen here. So we now have a single, it's going to be an earthworm.
Earthworm, yeah.
Yeah. So this is an earthworm zygote. Single cell. It just recently got fertilized. It's going to start dividing. Then it's going to turn into a hollow sphere of cells, right? At which point, how, in your words, does it go from a ball of cells into a tiny little baby earthworm? What actually happens?
Goes from what again?
A hollow sphere of cells into a baby earthworm. How does that happen?
Well, I don't claim to know exactly how earthworms.
Well, it's basically the same for all animals. So if you know how any animal develops, it's not too difficult.
No, it's not the same. So if you're talking about a fruit fly, it has a specific way. If you are talking about a human being, it has a completely different way.
The first step in all 3 of those animals is exactly the same. Do you know the first step?
I know.
I'm telling you now that a fruit fly, for example, will have an egg. The nuclei will split and will replicate, and then cell membranes will occur. I don't know how the earthworm works, but if it works like like human beings, cells will start splitting.
So at this point,
can I ask the question?
Yeah, you know what?
That will make it specialize, and then
why don't you go ahead?
Shape. Yeah.
Yeah. The answer is gastrulation.
Yes, gastrulation is in fact what I'm looking for. So here's what I'm concerned about, Ahmed. The things that you're talking about, like what kinds of information we need, what kinds of blueprints we need in the DNA, all of this requires you to have at least a basic grasp on things like embryology in order to know how anatomy develops over time, like how Hox genes work, how morphoproteins work, how different proteins affect the actual structure. And then by this, I mean even like structural proteins and proteins to do with biochemistry, how this will affect the actual structure. Because unless you know how those things work, you can't really talk about the genetics that it would take to go from any one morphology to any other. And so I am concerned that you are trying to wade into waters that you are completely unfamiliar with. If like the fact that you don't know, let me finish real quick, I'll be very fast. But the fact that gastrulation escapes you means that I don't think you know anything at all about the topic of how anatomy develops. And this is concerning to me because it seems like it's the basis of your whole argument. And now I'm done.
In case you aren't aware, Ahmed, you are still screen sharing. I'm telling you this because maybe you are like doing like maybe you're searching something.
Yeah. So I would actually like to keep screen sharing here. So yeah.
Okay, you're welcome to.
So
no, I was wondering. Like I remember all the time when I was screen sharing and I accidentally clicked on something I didn't want to show, and then oops, like okay, and I wanted to prevent that from happening to Ahmed. So
yeah, sorry.
Yeah,
thank you for that. So let's look at that. I don't know what's happening now. Just a second. Because
it's also interesting to like gastrulation is actually pretty interesting because the mechanism of why. Go ahead, sorry.
Yeah. So here we're looking at that. So you start with a cell. The cell was what I exactly said is that you'll start with a cell. The cells will divide, and then morphogens will tell the cells where to go and how to shape. And then you will start getting these cells to specialize, etc. Were you expecting me to give a full thesis of how morphogenesis works?
No, I would like a basic idea about things like how Hox genes affect things.
Actually, what I said was correct. If you wanted to go through the complete technical terms, fine, we can switch the discussion to embryology, but this is not what we're talking about.
But Ahmed, what you said was that every organism was different. But at the point that I was telling you about, all animal embryos do the same thing. They gastrulate. Now, granted, I skipped over division into different germ layers because that it wasn't really as important as like setting up just things like the very basics of how a Hox gene works, because whether you have 2 germ layers or 3, it doesn't really change things like the fact that Hox genes are going to set up gradients of certain chemicals that will then tell the organism along which end is, say, the oral end or the aboral end or the anterior or posterior. It will set up things like planes of symmetry, whether you get multiple planes of symmetry like with cnidarians and other radially symmetrical, or you get a single plane of like. Do you know how any of that happens? Like when I say a gradient when I say a chemical gradient.
I happen to do, but I'm not claiming I'm an embryology expert. But I happen to do to know the general mechanism, and what is important here is that for this mechanism to work, you have a directed build-up of what the animal is, and if this animal is going to have an eye, then the information for how to build that eye, regardless how complex that eye is there, and it is as complex as that eye wants to go.
Okay.
So we go back to the same thing.
What is the difference in information? This is actually something that's very well established. We can do experiments to determine whether or not this is true, and we can do experiments with embryos and find out exactly how this works. What is the difference in information between the fins of a trout and the fingers of a mammal? Do you know what the difference in information is?
What is the point of changing completely the organ that we are discussing?
Well, I would say that there's a pretty big difference between the fins of a ray fin fish and the arm ending in fingers of a mammal. They serve very different functions, but they're controlled by the same genes.
this is not my question. The question is, you have moved from something where you're essentially doing exactly the same shift of examples that I protested when Jackson did.
The reason
when he is taking things that are already at a peer-to-peer level of development, let's say, and you have already the components and maybe Hox genes, whatever other mechanisms are directing them. But now, when we are looking at the Cambrian explosion as an example, we are looking at very, very obvious changes between what was there in the Ediacaran and what was there in the Cambrian. Even I think that most of the creatures in the Ediacaran had only 2 layers of cells, if I remember correctly. And
we don't actually know that.
We have 3. I think statistically this was the case.
Well, actually, we don't have statistics on that because of a few reasons. One, because of heavy taphonomic bias in the Ediacaran, we don't actually know what things existed. We can't actually run a statistical analysis on the population.
Although we can make inferences, like we know that there were bilaterians before the Cambrian, and bilaterians are pretty much like triploblasts, and they have 3 cell or tissue layers.
So we know that triploblasts definitely predate the Cambrian. We don't know how far back they go. We also don't know whether or not rangiomorphs are in fact metazoans. They might not be.
I've seen pretty good arguments that they are, but of course that is still debated.
Yeah, that's the thing. I think that they're either they're probably either metazoans or they're stem metazoans, right?
Yeah.
Like that's reasonable, I would think. They're probably closer to animals than they are anything else.
I've seen arguments that they are closer to eumetazoans, like the jellyfish, the bilaterians, but they are more closer to us than they are to sponges.
I've seen arguments there too, but again, it's still one of those like we're not sure. We cannot say whether or not triploblasts outnumber diploblasts in the Ediacaran. I think that might be reasonable. That might be the case, but we don't know. And so. But the thing is, the reason I'm getting into this embryology thing is because the kind of genetics that it takes to tell you whether or not you're going to develop this morphology versus that morphology, which is what we're ultimately talking about, right? The difference between, an ant and you is a difference in your morphology, primarily. Sure, there's different biochemical pathways, but ultimately, if you had the same morphology as an ant, you'd basically be an ant. And the thing is that this gross morphology is controlled by a set of developmental genes that are shared by virtually all of the organisms that you can think of, and they don't generally change by adding new ones that didn't exist before. Usually all that changes is the regulatory signals that say, hey, when do you express and when do you not? So there isn't some blueprint. There isn't a picture of an eye sitting inside anything's genome. All there is a series of shared across wide groups, developmental genes that say, hey, if you tell these cells to fold, to divide, and to die in this particular pattern, just do it in this pattern. These cells fold, these cells grow, these cells die, these other cells grow, now these cells fold. It's sort of like an origami pattern. The eye will develop out of these cells. But if you have a different signaling pattern, you can get an entirely different eye, because interestingly, the genes that control say the tetrapod eye, like you or I have, are in most cases the same developmental genes that control compound eyes, like in fruit flies. And they're just different mechanisms, or they're just mostly just different patterns of how these developmental genes signal. There isn't some great, huge amount of extra genes that are added to go between, say, a tiny little insect and a human, or even their common ancestor, which was probably just the first bilaterian, right? And even between that, which probably had something like eye spots, but we know it would have had all of these same developmental genes. There haven't been a whole lot added. And while some organisms have lost some, like we know that water bears have lost some of their Hox genes that would develop into what in most organisms would be medial sections. They just don't. They kind of skip over that. But still, we have these. This is why Jackson put up that picture of the deep homologies in various Hox genes.
So, aside from deciding that a fin can turn into a limb by reprogramming how it looks like, which I have no problem with completely.
Well, it's how it grows.
Let me ask you a series of questions. let me ask you a series of questions for a change. Okay.
Okay. Sure. Go ahead.
Do you concede or not? Do you concede or not that building any organ needs a programming of the design of that organ? Do you concede that or not?
What do you mean by design?
How it will look like, how the outcome will be.
No, you do not need an idea of how the outcome will be.
This is actually a point I want to make.
To have something like an eye or a limb or a hair follicle or anything, this has to be programmed into the organism. Is this something that you concede or not?
Depends on what you mean by programmed. Yeah. That's the thing. What do you mean by programmed?
This is actually one of the things I want to point out. Like you are thinking like in terms of organisms, you're looking at things from the top down, like as if there is a blueprint that dictates the morphology of the organism. But many things in biology comes from the bottom up. They emerge from underlying processes. Like for example, if you look at the cell membrane, for example, which are a bit of lipids, right? Are you familiar with those? Like phospholipids, like one end is hydrophobic and the other end is hydrophilic. But if you put those stuff in just plain water, they will self-organize without any specifying information mumbo jumbo thing, or they will just automatically self-assemble into bilayers and vesicles and also double membranes, basically. So you don't need any specifying information to do that. And actually a similar thing happens with gastrulation too. Like the way gastrulation happens is just because when the ball of cells grows at a certain size, then it will automatically collapse into a gastrula spontaneously without any specifying information to say, oh, dimple here or dimple there. No, you don't need that. It will just spontaneously collapse into itself to form this structure. So you have processes like those that will determine the overall body plan, but you don't need any top-down blueprint design to specify those things.
So, in other words, I do contest that you need a blueprint or a design in order to make these things.
I'm asking a very simple question. You have an organ, okay. Let it be an eye, for the sake of the example. I'm not agreeing with what Nesslig has said, but
you should, because he's 100% right.
Yeah, because cell membranes are not phospholipids of one type that are autonomously folded. This is a hyper simplification, if not an oversimplification. But anyway.
I want to hear the. I want to hear. Hold on, Nesslig. I want to hear the end of the question. Let's hear it.
Yeah. So the question is, you have one organ, and that organ to form, do you concede or not that the specifications of this organ is built into the organism?
Not in the way that specification is usually meant.
It doesn't happen. It doesn't just happen. You don't. So an eye forms because what?
An eye forms because of signaling patterns in developmental genes. The patterns are specified, but the end product is not specified in a way that humans specify things. Like when we say, you specify the design of, say, like, a building, right? You have a blueprint, which physically is a representation of the actual construction of the end product. This is more like a recipe, where I say to you, okay, put these ingredients in this kind of pan and put it in the oven for this long and take it out. And I don't give you a picture of what's going to come out. I don't tell you what it's going to be. But when you follow the recipe, a cake comes out. It wasn't specified at the beginning in terms of like, this is what the cake is going to look like, this is what it's going to taste like. It's just a set of instructions that if you follow them, will end up in the end product of an eye. But at the beginning, there's no way for you to really know that beforehand.
For who to know? Now, actually, you have actually answered. You have answered so you concede that there are a set of instructions in the organism that unfold to the extent that an eye is formed. Correct?
Yes,
or not?
I will agree with that.
Okay. Good. And if those instructions are messed up, the eye will not essentially form. Correct?
No. Disagree.
It will still form if you mess up the instructions?
It's going to depend on in exactly what manner those instructions changes. In many changes to the instruction will simply result in a differently shaped eye or a differently sized eye or an eye with a different shape of lens or something like that. But it will still, in most cases, be an eye. There are very few mutations that have ever occurred in any organism that result in eyes failing.
It would be a different eye, right?
Well, it would be a differently shaped, or it might be a differently shaped eye, or it might be an eye that uses slightly different chemistry, but it would still be an eye.
We agree.
Nesslig, I'm not seeing your presentation.
I'm happy with this answer. So you agree that
I don't know why it show.
You have such bad luck with that in me. I don't know why.
So we agree that specific instructions.
You just cut out. You cut out. I'm sorry. I didn't hear what you said.
What's going on?
You cut out. Yeah, I need you to restate your last.
Yeah. So I'm happy with the answer. So you agree that you agree that a specific set of instructions will form an organ, and changes to this instruction will change the organ, regardless to the better or to the worse, or it's still an eye or not an eye. It is going to change.
Yes.
So you agree that the specific set of instructions that will give you an eye spot need to become a different set of instructions to give you the compounded eye of the trilobite, or I don't know what you call a pinhole camera for some other creature. Right?
Well, it depends on what you mean by set. If by that you mean you'll need additional instructions, not necessarily. If by that you mean that the exact pattern specified by the same genes needs to change, then yes. Because those aren't the same thing. We don't necessarily need to add new genes.
You have an instruction that specifies an eye spot.
No, you don't have an instruction that specifies
you have an instruction that specifies an eye spot.
No, you have a pattern of developmental genes,
you have
which taken together result in the development of an eye spot. You don't have a single gene that says build an eye spot. That's not how it works.
You have a pattern of instructions that builds an eye spot.
Yes, I can agree there.
A different pattern of instructions that builds a compounded eye or a human eye, correct?
Yes, I'll agree to that.
Which instruction set, in your opinion, is superior to the other one? An eye spot versus a trilobite compounded eye or a human eye? Simple question.
Whichever results in the most reproductive fitness. Which, in the case of if I'm living on land in reasonably temperate environments, the human eye. If I'm living under the water between corals and I'm mostly feeding on things like algae and my primary defense is hiding, then the eye spot actually probably is probably a whole lot better because it's less injury prone. There's going to be, I don't actually need really to see predators, so it doesn't require as much metabolic input. Yeah, eye spot would be a whole lot better. So it's going to be context dependent as to which one's better.
An eye spot versus a trilobite, both are living underwater for...
Oh, okay, sorry, yeah, you're right. You said trilobite. I'll forget what I said about humans. So in the case of a trilobite, where I'm mostly either living around on the surface and my main defense is actually running away coupled with a mineralized exoskeleton, and I'm doing things like going up and down the water column, occasionally swimming, stuff like that, then yeah, the compound eye is going to be better. But that doesn't mean that it's objectively better. It's better in certain environments, filling certain niches. That's one of the reasons why trilobites, in many cases, in independent lineages, lost their eyes. We have blind trilobites because the eyes were too expensive. They weren't worth it, so they got rid of them. So it's not a yes or no.
The question is not whether it's better. The question is whether you need a superior set of instructions or not.
No, you don't need a what does it mean for it to be superior? How would I know?
So the amount of instructions that are needed to build an eye spot are more or less, in your opinion, versus the amount of instructions that are needed to build a compounded eye of a trilobite?
I mean, you're going to fundamentally use the same genes, so you're not getting extra genes, but you are going to change the patterns in which they come on and off. So the signaling pattern is going to be a bit more complicated in the latter case, but it's not going to be necessarily have more information. Like, is it more information to say stay on for, 14 hours than it is to say turn off and on twice over the course of 13 hours? I don't know. I don't know how to assess that.
You need instructions to shape it. You need instructions to build all the different components, to connect them together, to put them together in fiber cable, to send them to the brain, to have corresponding brain. You need a system. So the system that specifies an eye spot that says on or off, or there is light or no light, versus the system that builds the corresponding thing for a trilobite? Which one has more instructions?
I don't know how to assess that because I don't know how to say.
Also, that's actually your job to do it.
Yeah, yeah.
You are, yeah.
You need to tell me whether or not more or fewer signal changes and signaling patterns. How do you measure these things?
You seriously don't know if just specifying an eye spot versus specifying a complete system of compounded eye is
and
not...
Let's say we are both ignorant of that. Like, how would you inform us about how many instructions it takes?
So if we go back again to the thing that I were sharing, you need loads and loads of more instructions to build the trilobite eye, which I was actually fascinated to show.
You're backing the question. You're saying you need loads and loads, but we are asking how many you actually need.
The question was for you, but you asked me. So
yeah, but the thing is,
I was pointing out that this is your job to show.
See, Nesslig and I aren't sitting here saying this is more information than that. You are. If you want to know how to know if there's more information, that's something that's on you. I don't care how much information there is because I don't have a good metric for it, so I don't intend to try to measure it. If you want to try to measure it, if you want to try to say this has more information than that, that's great. But it's on you to come up with an objective metric that anyone can use to determine which one has more information and come to, at least within a margin of error, the same answers that you do in each case. If you can't do that, asking me to do it for you isn't going to help because I'm probably just not going to. So I don't have a metric. For some reason, presentation isn't working, and I'm not sure why. Let me see if I can figure this out.
Oh, there it is. I can see arguments. Yeah, I can see.
Yeah, I can see it now.
I don't know. It doesn't look good. Yeah. Like I was just trying to show, if I may, I was trying to show a paper called "Is the Cell Really a Machine?" by Daniel Nicholson, who deconstructs the worldview, like or the view that the cell or the biology is like a machine or like a blueprint. But anyway, go ahead, Ahmed.
Yeah. So this is the trilobite eye. And it does have lenses, so you need to specify the material that is building the lenses. And it is essentially not even so it has a double structure. It has actually 2 lens layers, and it has focal points, and it is something that is all. And then you will need to specify how they get arranged. There are those separations between them. And then here are the things, okay, and they're arranged in a certain pattern. And let's say, of course, at the end of each of them, there will be a photosensitive material that will then transfer that back. So you need here, versus an eye spot, you need to specify lenses, you need to specify shapes of the chambers, you need to specify whatever is in the back, you need to specify the neurotransmitters, and you need to specify whatever is in the brain that will catch that.
Okay.
So that is 6 or 7-fold, not just one extra thing. And each of those are made of proteins, and each of those will have whatever.
The lenses aren't made of proteins.
The lens is just like a mineral, right? It's basically a rock, actually.
Yeah, as is the exoskeleton.
It's made of.
So we're already not understanding what trilobite eyes are made of. Which again, this is one of the things that concerns me. You'll jump into the details of anatomy of trilobite eyes without even knowing what they're made of in the first place. That would be the kind of thing that you would need to know how they develop morphologically and how they might have.
Let's say that the lens is made of a lipid. I don't really care. It needs to be specified.
Okay, but is a lipid?
The specification of a lipid is different than the specification of a protein.
Right. How do we know which one of these things has more information?
This is not the issue. The issue is whatever is making up this lens, and I don't know, and it doesn't make any difference whether it's a protein, a carbohydrate, or just
the lens is made of mineral. The lens is emitted from a mineral, and
so it doesn't make any difference.
Yeah.
For 5, hey, we got a super chat. I'm gonna read it. Wait, hey, we're reading a super chat.
It has come here.
Ahmad, we're reading a super chat.
It has come here because it has been specified, isn't it? How did it come here?
We are reading a super chat. Silver Harlow for 5 says, the recipe to make a cake might make a cupcake or a bundt cake if the instructions say to fill a pan which comes outside the recipe. Yeah, that is actually I mean, the recipe thing is not a great analogy, but it's not terrible either. And the thing is that, yes, that is the kind of minor change that can result in fairly significant morphological changes. You can go from, you know, one kind of structure to a very different structure simply by a quick change in exactly how the developmental gene operates. For instance, the difference between a scale and a feather is basically that you get a secondary expression of Hox genes that result in scales that go in a circular pattern. That's it. That's all it takes. You get a second expression of them in a circle, and suddenly you have the first kind of protofeather, which is a hollow filament. And yeah, that's basically the difference between scales and feathers, is a secondary and circular in pattern expression of a particular developmental gene. So yeah, and does that require more information? I don't know. I don't have an answer to that. And by the way, you guys should be able to unmute. I can't. It doesn't tell me that I can unmute you. So sorry, I did have to mute because we were getting too much over talk when I was getting to
No, actually, I muted myself.
Oh, okay. Well, I do want to say I don't think we're really going too much into this, so we can keep going. But I'm going to open it up to audience questions at this point. So if anyone has a question for anyone here, at me, say who the question is for, and then just type in the question. Of course, if there is a super chat, it will go to the top of the line. But we've got plenty of time. I don't expect that to be a requirement to get your question in unless it's very near the end. So, but the reason this concerns me is if I wanted to talk about the detailed specifications, say, of making a computer motherboard, right? But then I had trouble expressing what a logic gate is. You see how that would be concerning to anyone who is trying to take me seriously, right?
Yeah, but how does that come into context?
So the kinds of questions that you're asking are questions that ultimately go down to embryological development, because you're asking about how gross morphology occurs, and those are embryological questions. But you don't know what gastrulation is. You don't know how things like chemical gradients establish axes on an embryo. I'm not entirely sure you know the difference between triploblasts and diploblasts. Like this is like logic gate level.
I didn't gastration. It doesn't mean I don't care about it or I don't know about it. I mentioned that the cells will divide, morphogens will tell them how to shape and how to form. It will direct the specification of organs, and then you will have a complete system. And if this is a discussion about embryology, we can go into the details. This is not the issue.
All right, well, we have a question.
The issue is we are into a discussion of specification. Go ahead with the question.
We have a question, and this is going to be for everyone. The question is, what is the airspeed velocity of an unladen swallow? And of course, my question is, what do you mean, African or European? This is from Rodent, no last name, by the way. That was just a joke. Okay, yeah.
What is that? Okay, so the topic here, Dapper, is like this. Just my take on this. The more one looks into the theory,
mm-hmm.
and just look at the discussion that we were having, looking at an eye spot and a compounded eye, and having to have a discussion about which one of them needs more information to specify is sort of outrageous. And the reason it is this way is because you have a theory that is cornering the mental process. It is unfreeing the mental process of people looking at it.
I don't really know what that means.
So you are not free to follow the logical sequence of thought Because when you will say it is more complex, I do not think any researcher will really hesitate about it. But because the next
now you're again appealing to intuition, like, oh, it's obvious, it's more complex, so therefore it needs more information. Like, but of course, we are still asking you to objectively define it.
It is definitely from okay, it is more complex to have a compounded eye versus an eye spot. This doesn't even need proof. One of them is a spot, and the other one is an organ.
Actually, I would say it does need proof.
It's part of a complete system.
Real quick, I would say it does need proof, but I do think you could reasonably provide evidence that compound trilobite eyes are more complex. But more complex, in order to tie that to new information, you have work to do to get there.
Yeah, I don't, because one eye spot is corresponding to one, I don't know, cone or rod in a bigger eye. So you have all kinds of eyes, and every one of them is more complex than an eye spot. Yet we have to keep on discussing whether an eye is more complex than an eye spot.
No.
Because then the consequential thing will be that there is extra information, and then the question is, where is the information coming from?
Okay, hold on.
So
you're shifting from information to complexity, and those are different questions. Is X more complex than Y is a different question than does it require more information to build X than it does to build Y? So, for instance,
when things are more complex, they require more information. This is a given.
Not necessarily.
Oh, absolutely, it is.
I don't.
If you have an efficient system, describe something, so you take inefficiency out of the way, then more complexity means more information. There is no question about it.
Ahmed,
there is no question about it.
Let me explain this. Okay, so let's say
what do you mean it is not? What do you mean it is not?
Let's say I have a blueprint for a house. That house is less complex than the Mandelbrot set, because the Mandelbrot set is infinitely complex. But it takes far less information to generate the Mandelbrot set than it does to build a house, because all the Mandelbrot set is a simple algorithm that says, if for this complex number, this series results in an infinite number, it is not in the set. If it results in a number that is finite, it is in the set. That's all it takes. The Mandelbrot set is literally as complex as things can be, because it has infinite complexity at all scales. But it takes very little information to specify it. So it is not the case that more complex requires more information. Fractal patterns, as a rule, end up being extremely complex, but very simple to specify in most cases. You can make a complex to specify fractal if you want to, but infinitely many of them are very simple to specify. You can specify infinite ones just based on the Mandelbrot set, because you can specify infinite Julia sets based on it. So again, it is not the case that it takes more information to make more complex structures. That simply isn't true.
You have arrived to this conclusion because you have ignored half of my premise. So what I exactly said is that, setting efficiency of description aside, a more complex thing will need more information.
What is efficiency of description?
When you specify a Mandelbrot set, you specify it by writing an equation.
Well, it's an algorithm.
Because this is the most compacted way, and hence it is not a complex thing. It is a solution to an equation.
So more complex things will have more genes?
When you want to specify a house, you cannot compress the specification of a house into a single line of equation, which is going to unfold into a Mandelbrot set.
Actually, I can specify into a single line of numbers.
Is a mathematical algorithm, and it can run infinitely. And there is no extra information in the Mandelbrot.
I can simplify the house.
Whole information is included.
I can specify the plans for a house, or simplify them into a single number. It'll be a long one, but it's a single number, because blueprints for houses are expressible as images.
Why you can't?
Now, all images on a computer are saved as essentially a single number. You can take any image file in a computer.
Because you cannot give a single number.
You
cannot give a single number. You cannot give a single number to an engineer and tell him build the house for me.
Well, you might have to give it to a computer.
And if you say I will put the AutoCAD file that specifies the house in an AutoCAD file, and I will save it, and it will transfer to a set of bits, and then I will consider this a number made of 1 gigabyte of bits, then you are misrepresenting what a number is. This is a misnomer.
Well, no, I'm not saying the number of bits.
If we are going to do that, then what you will need to do is also to save the Mandelbrot equation into a file, and you will find that that file will be, I don't know, few bytes, versus the AutoCAD file for the house is few mega or gigabytes or whatever it is.
Okay.
So it will be obvious that the maximum compressible state of the Mandelbrot set is much, much more less than the specification of the house.
Okay, but then...
And this is exactly what I was saying.
How does compressibility...
When you are comparing information, you have to use the same numerator. Exactly. That's the whole trick.
So when
you use the same common denominator, you have to...
Okay, but I'm still not seeing how... Since we don't have any obvious analog for compressibility in organisms in terms of the genome, I don't see how this refutes the idea that more complex structure does not necessarily require more information. You need to tell me why that has to be the case.
You have returned to the very first thing that I have said, so I'm not sure if you are agreeing with me now or not, that since we do not have a good grasp on how information is being encoded into the whole genomic, I don't know, system, then the only way we can judge is to judge on the observed design. And we can also trace that back. We can also trace that back when the difference is so big, when you are just looking at an eye spot versus a complete eye, Dapper. And you know that the formation of that complete eye, and by the way, a lens, I just Googled it, and a lens, I was correct, a lens does contain protein, by the way.
What lens are you looking at?
In us contains
yes,
protein.
human eye lenses are proteinaceous.
Lenses, yeah.
But the protein...
But the lenses of trilobites are made of calcite.
And it's called crystalline. And I don't know if the trilobite eye had, I'm not sure if you do know, but if this is a lens, then I was not so bad to assume that it has protein.
Well, no one said like no lenses. No one said no lenses are made of protein. Trilobite lenses are not all lenses.
This is actually one of the reasons why trilobite lenses are like one of the first well-known lenses or eyes, because their eyes are made of rock, basically. Yeah.
So when I was saying to specify the lens, to specify the protein, I was not in error. But anyway, it doesn't make a difference really what you need to
because trilobite eye lenses are made of calcite.
The issue is to specify something, you need information to specify it. If you want to specify an eye spot, you need information.
I don't know, like if you look at, for example, a snowflake, it is a very complex shape, and the intricate shape that you have makes you think like, oh wow, there must have been a very large degree of specificity of information to specify this shape. But the shape of snowflakes emerges from the chemical interactions, basically.
A snowflake is a fractal, and if you can crack the equation for it, you can generate it with very small amount of information. So again, it goes back to this. When you are comparing things that are not of the same nature, then either you would be at the frontier of what that thing really is, so you can arrive to the exact level of efficient compression to represent it in the smallest amount of information, or you can't. Now, when you are looking into a biological being, and you are looking at a specific organ, and you see one organ that is a single part, or a very few number of parts, and you see a more complex form of that organ, like an eye, a compounded eye versus an eye spot. And it has different materials, it has different components, it has different wiring, and it has different output. This one says on and off, and this other one says you have a rabbit in front of you, or you have a fish, or you have whatever else. And I really cannot believe that we are discussing that one of them needs more information than the other.
Yes, because you haven't given us enough information.
It's because you haven't defined the terms yet. So it's
whichever metric. In this case, whichever metric, one of them is a protein, one of them is a chemical substance, and the other one is different cells, different proteins, a system that will make the morphology work, and the wiring of this whole thing.
Ahmed.
So there is no comparison whatsoever. So how come why are we dwelling on it?
Because it is a major point of your contention about evolution, that there is a problem about information. But as far as we can tell, you might as well be saying that there's a problem about, and then just making weird like fart noises with your mouth. There's a problem with, because you're not saying anything that means anything that is objectively verifiable. When you say, this has more information than that, and we say, how do you know? And then you give us some kind of hypothetical thing like, oh, if we could make it in a CAD program, and then I dig into that and I say, okay, well, does that mean that this is the case? Because based on your definition, it seems like it would be. You can't say yes. You then just kind of dissemble into another attempt at a different definition. At no point have you been able to give us something that we could use in all cases to objectively measure information content in anything, and then still agreed with yourself about it when pressed. Yeah, that needs to be the first thing you do.
Yeah. Oh, sorry.
It's like someone coming to someone coming to an engineer and saying, "Look, all of your engineering principles are wrong because you're not taking into account the wobbly factor of your materials." And you say, "Well, I've never heard of that, or I don't know what that is. Can you explain it to me?" And they can't. Then you don't pay attention to the crazy man talking about wobbliness factor, because he can't tell you what it is.
And when you're saying, like, why are we even discussing this? Then your answer basically becomes like, why aren't you agreeing with me? And that's not an answer.
No, I'm saying that we did actually do exactly what Dapper said. So when we looked at proteins, we find that proteins are more. When we look at cells, we find that this is just a single chemical sitting inside an algae, and the other one is a complete structure of different cells. When we look at morphologies, this is just a spot, and this is a complete formation for the eyes, arranged together, a neural link.
But the thing is, you didn't go from that to a methodology.
Each and every level of specification we have addressed, and at each level there is more information. So where is the dispute?
This is that's the thing. I don't know that there's more information at each level, because you haven't given us a metric to how to tell at any given level. That is a thing that we
we specified the number of proteins. So, we specified in this example the number of proteins, and they are more. The number of cell types one doesn't have at all, and the other has.
How do I compare between those two levels? So, okay, let's say that there's an organism with one cell, and it has 18 trillion proteins in it. And there's another organism with 4 cell types, but between all 4 of them, it only has 800 proteins. Which one is more complicated? Which one has more information?
This is not what we are discussing. We are discussing this eye versus that eye.
The whole point of discussing the eye, but the whole point of discussing the eye was because we were using it as an example to get at information, to get at what your whole point is, which is that information is a problem. But you can't tell us what information is in any case, because even when you just now gave your most specific answer, was talking about protein numbers and cell types, and I gave you 2 hypothetical examples and say, okay, if your metric is useful, tell me which of these 2 hypothetical organisms would have more information, and you couldn't answer that question. That means you do not have a definition that is useful to anyone for anything, Ahmed. And I would really like you to have one, because then we could actually talk about whether or not your information thing is a problem. But as it stands right now, no one can have that discussion with you, because you cannot get to step 1, defining your terms.
Maybe you're not giving me the opportunity to add.
I mean, you've talked a whole lot during this entire time and during the previous 2 hours. And at no point did you give an answer that when I then asked clarifications about, you didn't go back on. Find an answer that when I say, okay, if that's true, how do we use it? And you don't have to reverse and say, well, we can't tell in this case.
I give you an answer to what are possible measures to compare the eyes, and then you go into a hypothetical example. I cannot answer to a hypothetical example. Give me 2 real organs and 2 creatures.
Real organisms.
We study them.
Which eye is more complicated?
No, real organs.
Real organisms.
About real organs.
Yep. Okay, real organs on real organisms. Is that okay? Okay, which eye? Well, lineage. Everything's on the same lineage, so it doesn't matter which 2 organisms I give you. That's the whole point of common ancestry.
Not everything.
Which eye is more complicated? The eye of, say, an eagle, or the eye of a let's go with mantis shrimp. They're both bilaterian, so don't tell me they're not on the same lineage. What counts as the same lineage?
They are not ancestors.
No living organisms. No living organism now is ancestral to any other ones. So if you want me to go with organs that we can examine now, the only options I have available to me are living organisms. You tell me how closely related 2 organisms have to be to count. Tell me.
Also, I don't know the why hypotheticals are a problem, because if you have a definition, then hypotheticals would not be a problem.
That's true.
At all. Like, yeah, right.
So according to the theory, things are coming from unicellular organisms. So a unicellular organism with an eye spot is definitely on the lineage of a trilobite. Do you agree with that or not?
Not all of them. One group of them would be the most recent common ancestor of trilobites and some other group. So there are unicellular organisms that are photosensitive. But again, that's not any of them. Many unicellular organisms that have photosensitivity are not going to have been ancestral to trilobites, but some of them were. And so when you say they're not on the same line, I don't know what you mean, because everything is on the same line if universal common ancestry is true, which is the whole point of, well, universal common ancestry, which you want to argue against. So when you say these 2 things aren't on the same line, you have to tell me what would count as being on the same line. And if we're trying to use extant organisms, which we are, because again, that's where we have the most information, obviously. And if you can tell when things have more or less information, then me giving you things about which you can have maximum information has to be better. Because what it sounds like you're doing is relying on things we don't know to insert your uncertainty and then foist it onto other people. And we have a $5 super chat, which says, if you can't apply your metric to a hypothetical, then your metric is useless. From Rodent Low Last Name for $5. Thank you very much, Rodent Low Last Name. This guy is paying for my dinner.
And it's also funny, like the hypothetical was much more easier to answer than the example of the mantis shrimp and the eagle eye.
Yeah.
Right.
Like every time I try to get an actual example where you can tell me where there's more information or less, in a case where it might not be something that makes you feel fuzzy on the inside, because we're trying to get away from intuition. Intuition doesn't help in science. At most, it can tell you what questions to ask, but can't help you determine answers. That's why we're going to these examples and hypotheticals, because they can tease out your definition if you have one. But when every single example that I try to say, how do you tell if this is more complex or has more information than that? You can't give me an answer. All you can say is, I think that the eye spot is more complex than or more information than the eye. But even when we dove into that and we started to get into the embryological development of how do you know whether this is more complex, all you could say was something like, I think that, I don't even know, needing to specify more steps in a plan means more information. But even then, we weren't able to actually know by how much, how we could measure it, how we could tell whether it's more genes, because it doesn't always require more genes. At no point was there something that a scientist could look at and say, yes, I can use that as data.
Yeah.
Can you generate any points of data about information on anything that are objective? Because I've been trying for nearly 2 hours. Did you?
I actually did pick 4 measures, and on every one of the 4 measures, a trilobite eye is obviously more information rich than an eye spot.
But that's the thing; it's not obvious because I don't know how to compare these things.
Like now is he basically doing the argumentum ad nauseum. He's just asserting over and over again. It's obvious, but it's not obvious.
We counted things on all 4 levels, and on the materials needed in terms of proteins or any other substances, the trilobite eye has more, which needs more specification.
Well, actually, the only
on the cell types, one is no cell at all, and the other is multiple cell types, at least having cells that are building the shape of the eye and cells that are on the neurological side. Yeah?
What do you mean no cell at all?
And then
when did we ever talk about an eye, any kind of eye thing that didn't require a cell? I don't. You lost me.
Are you saying that a compounded eye is one single cell, or I'm not sure?
I'm saying you said that in one case we didn't need any cells. I don't remember that being a thing that we ever discussed. Was eye spots without cells?
Eye spot not a cell, yeah.
Well, it depends on what kind of eye spot we're talking about. If we're talking about eye spots on, say, a flatworm, then yes. If we're talking about eye spots on, say, a tardigrade, then yes. If we're talking about a rhodopsin protein on the cell membrane of some, bicont, then no, it's not a cell in and of itself, but you still need a cell in order for it to function. So you're at a minimum 1 cell, or it's not going to do anything.
Well, fine. I will take 1 as an answer, because we will not have fractions of cells. But then on the other side, you have multiple cells, at least cells that form the eye, and cells that form the neurons that take the signal. At least you have that. Plus the cell that is whatever is photosensitive there. Okay? So you at least have 3 versus 1, and then you have to connect them together, and then you have to have the extra information for the creature to know how to use this whole machinery.
And we have
on all 4 levels. You have a higher level of information.
Nope. You have a higher level of complexity. I'll agree with you there. But again, to go from more complexity to more information requires an argument. But we have a question from the audience. And this is from CRISPR-Cas9, who says, this is from CRISPR-Cas9, and he says, how can you possibly say the number of different proteins needed for 2 extinct animals to develop eyes? And actually, I would argue that generally speaking, unless they're very recent, very recently extinct organisms, you can't actually know, which is one of the problems that we're having here. Not only do we not really have a great definition of information, not only are we conflating it with complexity, but we're also
complexity is also not defined.
Right. But we're also putting it into situations where we have huge lacunae in the data. So all of this, it seems, and I got to be honest, now, Ahmed, on a personal level, I like you. Like, we have good interactions. We have nice chats. Usually off air, we have some nice chats. We'll talk about coffee and stuff like that. So please don't think that I'm trying to be mean here. But what I've gotten today for the past, we'll say past 4 hours, because we'll just take out that middle hour in the chunk and just kind of toss that out, has mostly seemed like intentional retreats into areas about where we have little to no data, coupled with sophistry to avoid having to define terms in a way that allows us to actually have real detailed analysis of organisms that could confirm or disconfirm your ideas. And what I would
it happens Dapper, that the Cambrian is an era where there is not that much data, and it is the topic of the discussion.
I mean, that by definition, although to be fair, the discussion very quickly went from anything to do with the Cambrian per se to simply information, and we couldn't even get to the point where we can say whether or not information can increase or decrease in a materialist way.
Or whether it really is a thing.
Right, we couldn't even get to the what is information. You couldn't tell us whether or not an onychophoran has more information than a spider. You couldn't tell us whether or not, even in cases where you say, I'm very confident that this has more information than that, when we drill down into it, the metrics that you give end up not actually being applicable in any objective sense.
What is remarkable about Cambrian Explosion is comparing it to the previous era, not comparing it to itself. Well, because this is why
Jackson pointed out is that we actually have like precursors in the Cambrian, and now we have also things that happened after the Cambrian. So it's like, it's not like boom, everything was there. It's like a gradient of like, oh, more complex things, more complex things, more complex things. Oh no,
except that we're discussing the Cambrian explosion in specific. You will have similar problems if you look at some million years later in the where mammals pop into existence or evolve or whatever. So the theory is full of those.
We've also had pre-mammal traps. It's also like
so we are taking one specific specimen of time here.
We are coming up on 2 hours. So it is. We're gonna have to call it. Unfortunately, we're probably not going to get any resolution in any finite amount of time here. So I'm going to say first, I want to thank both of my guests. Nesslig, I definitely don't have your channel listed in the description, which I apologize for.
It doesn't matter. I don't do much on my channels.
Okay, fair enough. But if I remember correctly, and I'm going to double check now, both Jackson, who was in the debate but who was not here, and Finding for Truth, they are tagged in the description, which yes, they are. So please go take a look at their channels, subscribe if you like what you see. If you, for some reason, came to this without having seen the original debate, that is also linked in the description, so please go ahead and watch that, because, of course, it's providing a lot of context for a lot of this. I hope you guys had fun, and I want to give a special thank you to Rodent No Last Name, Silver Harlow, Vandalia1998, and Vandalia1998 for your super chats. Your generosity is always appreciated. If anyone else does want to support me, there are, of course, links to help do so. And I'm going to start with Nesslig20. Do you have anything that you want to promote? Anything upcoming, whether it's you or anyone else? I will take your silence to mean no. Oh, wait, you're muted.
He was muted.
Do you want to promote anything? Anything that's upcoming for you or for anyone else that you want to promote? I'll still go with no. Ahmed,
I'm sorry.
Do you have anything that you want to promote?
Yeah. Okay.
Do you have any upcoming debates or anything like that?
Well, first, I would like to thank. I would like to thank you for the moderation.
Oh, you're very welcome.
I think it was nice.
I try to be fair.
Yeah, and the second thing, something to promote. Well, I would like to invite everybody to go to the channel, and I really like comments and proposing topics. And the channel is about what science is about, and what religion is about, and what science can benefit us in terms of finding out whether we're here by chance or we're here by design. So the other aspect of it is religion and is existential questions. So I would like to invite people there. Okay. One thing that I'm going to do soon, put together a comprehensive sessions about short things about evolution, starting from Luca to the human being. So maybe that is in a couple of months or something. Small ones, like series. Okay. I have already things, but big ones. But taking the gradient from the beginning until the end.
Well, don't be too shocked if you see some response videos. There might be some
I have no self-promotion.
Yeah.
Sorry, go ahead.
I can promote Jackson a bit because I am one of his editors for his videos. So go watch his videos, and then I will be happy.
Yeah. Basically, you're supporting Nesslig20 by watching Jackson's. In fact,
Jackson, by the way, I saw them to see what he has to say about the Cambrian before the debate, and they are fun videos, actually.
Oh yeah,
so,
Jackson has good content.
Since he's not here.
All right, well, thank you very much, everybody. I just want to say Tuesday will be Eric with Erica, and then I'm not sure which video is coming out on Thursday. There are a few options. Saturday is going to be another marathon debunking stream because we didn't even get close to finishing with that video that I put together. So we're just going to plow through ahead with that because I'm still angry at Genesis Apologetics. Basically, I'm going to keep doing those until either I run out of that video or I stop being angry at them. That's basically what it's going to come down to.
Basically, waiting for the death of the universe.
Yeah, basically. So
Yeah.
And then the Tuesday after that will be the next Kent with Bent, in which Benthoven will be wearing his pink tutu. So, yeah. So if you wanted to see Benthoven wearing a pink tutu, tune in. He'll be doing it again. And I want to say thank you to everybody for coming. And that's it. I hope all of you have a wonderful rest of your weekend and rest of the week. I hope to see all of you on Tuesday.